All right, welcome back. So we're on to the next part of the cell, which that is the nucleus. And sometimes, they'll draw the nucleus like in the center of the cell. It's not necessarily in the center, like this one's off to the side. But the nucleus is where your DNA is located, and it is that control center of the cell. So its function, it is to control all the cell activity, and it's to store your DNA. So inside it, you have chromatin, and chromatin all balled up are what your chromosomes are. And so it's long strands of DNA. And so as a human, we have 23 pairs of chromosomes, and they are inside our nucleus. They're not in the center area here, they're on this outside, and that is where all your DNA information, every cell in your body has this chromatin. And the functions of that chromatin is to has the information to make proteins. So we're not going to go into, because this is not a biology course, all the ins and outs on how proteins are made, how the amino acids are put together, and how mRNA and transferrin RNA can get quite complicated. But just understand that how the blueprints of how to make proteins are actually in your DNA. So you get ready to build a house, you have this blueprint. Well, the blueprint of the proteins that are going to get made for your body are stored in your DNA in these chromosomes, also called chromatin. But in our nucleolus, the center, this one's kind of off to the side here, the nucleolus, that is where ribosomes are being made. Ribosomes are a really important part of your cells, and those ribosomes are being made in the nucleolus. So the reason I'm having you watch this video, this will also help you in lab for some of those questions. So here's just a picture of a stain of a nucleolus. You can see it stains darker. All right, we're going to move on out to, and usually around the nucleus there's these another structure called the rough endoplasmic reticulum, or the rough ER. And so our rough ER is going to receive the ribosomes that are getting made in the nucleus. The ribosomes get made in this nucleus, and they'll get out of this little pore, and they call it out of this nuclear envelope or nuclear membrane here, and they're going to need a way to move around to different parts of the cell if be. So they basically attach themselves to this little highway system. So it's a tunnel system that transports proteins making. Did talk about this on the cell video for lab, so it's a little bit of a repeat here, but that's a good thing. We can always repeat it. Okay, so I paid for this little PowerPoint that we're clicking through. Somebody made it and it's really cute. I paid teachers, so give them shout outs for that. So just want you to watch, and we're starting in letter A here. That is where the nucleolus is. Here's all our chromosomes. We've got these pores. Here's an endoplasmic reticulum. And so in there, we're going to see the ribosome get made, and then all of a sudden it'll go through this little maze system. Some of them get stuck in the endoplasmic reticulum and they'll stay there. These are called attached ribosomes, and some of them will actually get out here in the cell. But so you can see what's happening here. So it's like a little tunnel to get transported. There is the endoplasmic reticulum. So we have another endoplasmic reticulum, but this one is smooth ER. There's no ribosomes. What's making that one rough looking is the ribosomes, but they do have a completely different function. This one's a tunnel system. This one actually, those lipids that we talked about on the macromolecule video, they help break down those fats, and it also helps break down toxins for your body. So the smooth ER has a completely different function going on with it. So it's not part of that little highway system. All right, so then we talk about the ribosomes themselves. So the ribosomes, they're a big player, and I think I said on the other video that I had a high school student said here they call it ribosomes, because ribosomes It's really ribosomes. Their function is going to make proteins for the cell. So again, you've got those instructions in here, but these little ribosomes are starting out made in the nucleolus. They come out of the nuclear pore. They're going to end in this endoplasmic reticulum, and let me go ahead and get transported by the rough ER. And their function is to make proteins. So some of our proteins for our body are getting made right in this area, and some are going to get made out here where these the ribosomes have actually come unattached there. So these are attached, and these are what we call free ribosomes. They're little dots in our structure for lab. So you can see them there. So here's what they do, because they're making proteins. The monomer for proteins are amino acids. So what they'll do is they'll get a certain pattern. And how do they know what pattern to put them in? Well, they get some instructions like ACC, CCGC. It's kind of some sort of magic code, which is called a codon. And again, we're not going to go into all the protein synthesis, but proteins are basically just amino acids that get stacked together. And so the ribosomes help do that with the help of messenger RNA. And they'll stack those amino acids together. Some proteins are just 5 or 6 amino acids. Some are hundreds of amino acids stacked together. And then together they come and make a protein. And then some of the proteins will be needed in your body, or I'm sorry, not in your body, but in a specific cell, or some protein might be needed for like 4 cells down the way. So sometimes the cells will stay inside, the protein will stay inside one particular cell, but sometimes the protein will need to be shipped to another cell that might need a protein. And so that's where the Golgi body comes in, or the Golgi apparatus. Okay, so this is really the Golgi apparatus here. These little bubbles that you're seeing here are called vacuoles. Actually, they're called vesicles. And the difference between a vacuole and a vesicle, vacuoles just hold things. Vesicles, like a vehicle, are going to move. We're going to see these little guys move here in a minute through the animation. So what the Golgi body, think of UPS, it's going to pack up proteins, and if they need to be shipped or changed in some way, they'll kind of modify them, maybe put them in different packages, and they're going to ship out for the proteins out of the cell. So they release in a protective thing called a vesicle, and that's what's going on right here, that little bubble. And we're going to watch the animation here in just a second so you can see it. Alright, so here we got the nucleolus. That's where we're going to start that. Again, our DNA is in the nucleus part here where the instructions are, but that ribosome is going to get made there. So we're going to make the ribosomes. These are the protein shipment steps. You've got 1 through 4. You'll be putting those in order on your Unit 2 test. Okay, so nucleolus is making the ribosomes. The ribosomes travel through the rough ER, and they make proteins as they go, because that's the job of the ribosomes. And so now the ribosomes have made their protein. Let me get my clicker going. And so that protein might not be in the correct order, might need to be sorted out. So a vesicle will take it to the Golgi. So here we're arriving at the Golgi. The Golgi will sort and package up the protein. See the animation of A4. And now this is the plasma membrane, which is surrounding the cell. It will release the vesicle out of the cell by exocytosis. And so we're going to see it leave the cell there. So these are our 4 steps. You got to make the ribosome. The ribosomes attach themselves to the rough ER. They make proteins. Those proteins will end up getting out of vesicle, going to the Golgi. Golgi will also then package up and send it out through a different vesicle. So that's the steps of protein synthesis. See you on the next video for the mitochondria.